introduction. So welcome to project presentation 4. Thank you. I know a few of our colleagues are coming in from that quick break, but I do want to take a moment to tell you that we will be hearing next from, and please, again, I am so sorry for names. I didn't have a chance to touch base with everyone on pronunciations before, so I apologize. And please do correct me when it's your time to talk if I mispronounced it. But we will hear from Yoshie Hanzawa, Bruce Hajek, and Xiaohan Kang on a project titled, I'm sorry? Oh, no, on a project titled On Modeling the Circadian Clock Gene Regulatory Network in Soybean. And I believe that Yoshi, you are kicking us off. Is that correct?
Yes, I believe so. Thank you very much for the introduction. Let me share my screen. Can you see my screen?
I can. I see you putting it in presenter mode. It looks great.
Yes, perfect. Let's see. No pen. Pointer. Oh, where is the pointer? I don't see pointer. All right. So while I don't have a pointer, that's probably okay. Arrow. Let's use a arrow.
You've got one. It's just very, very small.
Very small? Okay, well, that's okay. All right, so my name is Yoshie Hanzawa. I'm assistant professor at California State University, Northridge. Can everyone hear me?
Yes.
Okay, perfect. All right, well, so let me, how can I move my screen? Here you go. Okay, so the goal of our study is, here's a big goal. We'd like to understand how crop plants respond to environmental changes at the gene regulatory network level. So this is a big goal, but the environmental changes that is very severe and happening now, and economically crop plants that are less understood, but we really need to understand it. So instead of just looking at plants, if we can come up with the models of gene regulatory network, that would be extremely beneficial because I think we can make mathematical models to understand these networks of the gene hierarchies. If we have mathematical models, then we can basically understand the agronomic trait, important trait, and predict plant responses in different environment, and even pinpoint what are the key genes for effective modifications, such as genome editing. Also, if you have mathematical models of the gene networks controlling important agronomic trait, then we may be able to incorporate variation, natural variation, genetic variation in gene regulatory networks, and we can use it to predict plant evolution at the gene network level. So my goal, well, in my mind, is huge. And to look at that, we are looking at tiny parts of this big gene network. This is Arabidopsis model plant gene network controlling flowering regulation, reproductive transition. We are looking at this part of a subnetwork, Circadian Clock. Circadian Clock genes are well known to affect important agronomic traits such as flowering, many physiological responses of plants. And Circadian Clock gene network is very well characterized in model plants such as Arabidopsis thaliana. So we have a ground truth network. But here, the goal is to understand the crop plant. Soybean we are currently using. This is a major source of protein and oil, and there is a significant similarity in a set of genes and the gene function to the model Arabidopsis thaliana. So that is a great starting point. Now, let me introduce a little bit about circadian clock. It is an endogenous biological cycle that roughly repeats every 24 hours. It is ticking in each cell. It's entrained by environmental input, so it is ticking, but it responds to environmental input. It's comprised of generally 3 major parts. First part is the input pathway from environment and entering the clock. And the major part is a central oscillator, usually consists of a handful of genes, transcription and translational loops. The third pathway is the output pathways. Output pathways control multiple physiological, developmental, cellular events of organisms. In the case of plants, of course, development, flowering, hormone signaling, metabolisms, and biotic and abiotic stresses as well. So specifically, plant circadian clock is interesting. It is unique evolutionarily. Compared to animals, it has a different set of genes. So it is considered to be a different origin evolutionarily. But all of those clocks in any species, in any domains of the organisms have interlocked feedback regulation loops of circadian gene expression. This is a simplified, a little bit older model of Arabidopsis circadian clock. It has a core loop structure and morning expressed genes loop, and then the evening expressed genes loop. So this is a general structure of Arabidopsis circadian clock network. Most of the studies, as I keep mentioning, have been done in Arabidopsis thaliana using genetic and molecular approaches. And also there are multiple mathematical modeling attempts, let's say. So that's the reasons why we thought circadian clock network is a great material to get started. But Arabidopsis model has been developing. Now, well, we have more genes incorporated, and the basic structures basically is the same: morning, core, evening, but there are more genes. And in terms of regulatory interactions, we know that it mostly consists of inhibitory regulation, repressor regulatory interactions. This is very striking and specific to plant circadian clock network. It's on Arabidopsis. Now, then how about the crop species like soybean? We know that it's controlled agronomically important trait, but we really don't know what structure in crops the circadian clock network may look like. What we know is the individual genes homologous to Arabidopsis and individual genes functional studies. So that's why we wanted to know the network of soybean circadian clock. So we have to start with very much scratch. So our approach is basically start with network discovery. We don't have much information, but we can do RNA sequencing, taking the time series samples. And then we can come up with algorithms to use this RNA sequencing time samples to infer networks structure of circadian clocks. And based on that structure, we can build hypothesis. And then next step, test experimentally. Experimentally verify infer the clock gene regulatory interactions. And also we can experimentally identify and expand with new regulatory interactions. Once we do that, we can go back to the original network structure we initially came up with, revise, remodel, and come up with a new hypothesis that should be tested by experiments. Once the model becomes a certain level, then the next step is mathematical modeling. We can do further Ode modeling, we can do network motif identification and parameter identification. And the modeling, of course, basically gives us information, what is the data structure of the network and the new hypothesis that to be tested by experiments. Okay. So we first did the building this algorithmic package called CoSNet for causal inference of gene regulatory network. Thanks to Bruce and Xiaohan, it incorporates sparse linear regression, but I'm going to pass the detailed explanation to Bruce and Xiaohan. This is available from here, so please check it out. So this takes gene expression data, RNA-seq data. So we designed expression data as expression study. So we use the multiple photothermal light regimes and temperature conditions to perturb the networks. We use the first 25 degrees long day conditions, longer day lengths for 3 weeks. Also, we had a separate experiment, 25 degrees, short day, 3 weeks. Those samples after 3 weeks, we moved the condition to either higher temperature or lower temperature, keeping the same photo period, or the higher or lower temperatures and change the photo period from long day to short day, because soybean responds to short day to start producing flowers and responding to the short day photo period to initiate many developmental and physiological phenomena. As a control, we had a 25-degree short day, kept short day and higher temperature and lower temperatures. To genetically perturb, we use the multiple genotypes. Let's say this is a wild type typically used, although it contains a mutation in one of the genes that are controlling the circadian clock. This is, let's say, the all functional loci, and this one contains the E2 recessive mutations. This is a part of the gene in the clock. And this line contains E3 mutations. This is a phytochrome, so photoreceptor mutant. And then we took the time series sampling. This goes, we did extensive sampling actually. This is a day 0 before the condition change, and after condition change, we took the samples once in a day, which we haven't actually used for this particular project yet. But also day 0 and day 5, we took that sampling every 4 hours within the same day, 24 hours, so that we can see the oscillation of clock gene expression. For now, but we just started to use this data over day 5. But actually, this is a previous work, earlier work. We first started to use only the 3 time points, and we tried to see what we may be seeing. So this initial work was focused on larger network of flowering gene network that contained, we included 74 selected genes in this inference work. These include 14 clock gene homologs, only 14 actually. And we took only 3 time points per day. And the results of clock genes looked like this. We only used 3 time points, so not much of a resolution, but we managed to comprise of short day and long day networks. We found that among those 74 genes, those 14 clock genes really connected really strong and created an interconnected subnetwork. That's what we are looking at here. We don't get into too much of these inferred networks right now because this is only 3 time points. And although it is interesting, especially that many of the regulatory interactions we see here are activation as opposed to repression that we know in Arabidopsis clock network mostly have. So we moved on. We probably should use more time points. So we started to use 6 time points, and we did the same with the run COSNET. We ran the COSNET 9 photothermal conditions, 3 photoperiod, and 3 temperature conditions. And we found the first is that it seems like temperature has a much more strong effect to the structure of inferred networks, more than photoperiod. So here we are looking at combined networks of long day, short day shift at 25 degrees, 16 degrees, and 32 degrees. In total, for this work, we used 40 clock gene homologs, including the set of the genes we omitted in the previous work. So this is a more complete set of genes. We also incorporated slightly different algorithms in the COSNET package. COSNET has a modular structure, and we wanted to try how the different algorithms may behave. Overall, we saw similar structures, but somewhat different. So the next step, what we want to do with these 9 networks, it's complex. So we've decided to focus on 25-degree networks and looked at only the intersection of those 2 algorithms output. And we have much more simpler, well-supported network here, although it's still inferred network. The next step, we really need to do verification if we are looking at real regulatory interactions or not. So the first step, we selected actually multiple genes that are inferred as regulators and subcloned them and then subjected to experimental verification. Here, I'm going to show you only the 2 genes. LCL3, this is a combination names of Arabidopsis 2 genes, and LHY and the CC1 genes, but they're very similar, and we are using LCL for soybean name. And the PRR gene, PRR593, there are multiple homologous here. So the way we developed for the experimental system, it must be quick system. We can make transgenic soybean, but it takes multiple years. So we developed protoplast transgenic system, and the transfection rate of the transgenic expression of our gene seems to be very high. It's about 50 to 90% of the cells can express our gene of interest. Our genes are tagged with GFP so that we can see their expression, and we can use the GFP antibody to cool down those proteins. The first experiment, we want to know those regulatory interactions really regulate inferred downstream genes. So we did a qPCR analysis of inferred target gene expression. Second, we did a cheap qPCR experiment using the GFP antibody, pull down and look at what fragment we actually pulled down together. So that is to remind you those two genes we focused on. The one of the gene, here 593, and protoplast cells we expressed and took the time course and 6 time points we have. The cell line that's expressing PR593 expresses this gene highly, which is great, and the control cell line. Similarly, we expressed in protoplast cells LCL3 gene, that's another targeted gene, again highly expressed, much higher than control. We looked at the inferred downstream genes. In this case, we looked at mostly in this PR593 expressed cell lines upregulation of downstream genes. So we can probably conclude PR593 actually upregulate LAX and L4 at the predicted downstream genes. In the case of LCL3, we looked at difference in gene expression, but it turns out that it seems to be replaced compared to non-transferred cell lines, which is actually interesting. So it seems like LCL3 downregulates PR3 and PR5-9 genes. It is interesting because it's opposite to our inference results. This one seems to be correct. Now this one, opposite, but turns out the same regulatory interaction with the Arabidopsis. So that gives us a certain level of confirmation. Now, how about direct interaction or not? That we tested by chip, PR593. We designed several primers in different promoter regions and took the 2 time points. It seems like at time point 3, this high enrichment is statistically significant, much more so than time point 5. So we have a direct binding of this PR593 proteins. Similarly, LCL3 by chip experiments, we managed to show downstream genes are actually highly enriched, so which actually is great. As a summary, we managed to verify part of the inferred regulatory interactions by experiments. So we are confident that the de novo network inference approach is a tool, powerful tool to target lesser known systems to develop network models. Currently ongoing approaches, expanded effort for experimental verification and experimental identification of interactions by chip sequencing is currently ongoing. And we want to see the condition-specific interactions and mathematical model part is currently ongoing. So with that, I'd like to pass it to my collaborator, my modelers, Bruce and Xiaohan. Let me switch.
Hello. Thank you, Yoshi. I'm Bruce Hajek. So Xiaohan and I have been working with Yoshi for about 5 years. We're from the electrical engineering, communications, modeling side, and Yoshi's been teaching us lots of biology over the last 5 years. And I can say we're totally enraptured by the complexity and the beauty of the biology, but it's also a challenge to model it and have accurate models with all the experimental difficulty. And so we've seen end to end how we go from the data and preparation of samples to the graphical models. So with that, I'd like to turn it over to Xiaohan.
Okay, I'm going to share. Can everyone see my slides?
I can.
Good.
Yeah.
Okay. So thanks for the. Oops. Can you still see my slides?
Oh,
Sure
good.
can.
Okay.
Yes, it's in the beginning.
Yeah. I'm Xiaohan Kang, and I'm going to continue from Yoshi's explanation on the experiments and the overall objective, and I'm going to talk to you about the circadian clock in soybean from a network inference perspective. So in the next 15 minutes or so, I'm going to explain how we model gene regulatory networks. Basically, it's graph models versus ODE models. And I'm going to spend most of the time from the graph perspective, where I'm going to explain the COSNET package that we developed for the de novo RNA-seq time series data. And I'm going to explain to you what we call the motif mapping is when you have prior knowledge from other species and other experiments. And last, I'm going to touch on ODE modeling part. So a gene regulatory network is usually defined to be either a graph like this, or it can be defined in a more detailed way by differential equations.
You can't see the graph, Xiaohan.
You can't see the graph?
No, I just see the cover slide.
Oh, okay. Let me disconnect. Okay, I'm going to use my desktop.
Okay.
Can you see the slide now?
Yes.
And can you see it moving?
Yes.
Okay. All right. So I have 13 minutes. The GRN can be described by a graph like this, or it can be described by a group of these differential equations. And for the graph model, we can see that when you have a fixed number of genes, there are only finitely many graphs, because for each pair, you either have a activation edge or repression edge or no edge. But for OD model, it's much more complicated. Even a single graph can give you infinitely many OD models. And that's because you could have all different types of functions for the input function. For example, for the two activation edges, they can act as an AND gate or OR gate, or other kinetics in between, or you can also consider cis regulatory modules to divide the input into different groups. So that gives you a lot of complexity. There could be intermediate variables like the protein concentrations in different parts of the cell for each of genes, and there could be protein complexes and others. So that's going to increase the number of variables in the vector X. And there are parameters for these functions. So basically, the ODE models are very complicated, and learning the ODE models can be very difficult because you may not have enough data. And compared to that, learning the graph can be much more feasible because there are only much fewer candidates for the graphs. But then we still need to be careful about the term the graph, because when you are given a system, it could be possible that there is not a unique graph. So we have, as we have shown before in our paper for differential equation model, it could correspond to different graphs in different parts of the plant. So we need to assume, we need to make the assumption that there's a unique graph for the whole system in different parts, and that we have seen that that's true for many popular class of ODE models. So now that's the assumption, and we just want to figure out what the unique graph is for our plant, and we develop this COSNET, which uses algorithms based on sparse linear dynamical models that Yoshi just mentioned. One feature of the COSNET is that to assess the reliability of the predicted edges, we do what we call the perturbation analysis, which is to perturb the original data randomly and slightly, so that we might get slightly different prediction of the network. And then we look at which of the edges appears in most of the perturbed results, and that appearance give you a certain reliability score. But now the problem with the COSNET framework is that it's only using time series RNA-seq data. It doesn't take into account the prior knowledge from, for example, the other species like Arabidopsis, and doesn't take into account other type of experiments. And also it ignores what you are expecting for the network. For example, for the circadian clock, we expect to have a certain negative feedback loop in the main part of the network, which could be this two gene loop with an activation and repression, or it could be this so-called repressilator with 3 genes repressing one another. So we expect to see these features in the network, but COSNET is doing this in a de novo way, so it doesn't take these into account. So to take this into account, we propose motif mapping, which is to locate these motifs from the graphs taken from the prior knowledge. To be more specific, when we take a particular motif M, which could be this 2-gene network, then the 2 edges are assigned a weight of +1, -1 because of the activation and repression. So this motif is a graph with weight 1 or -1 on a few of the edges, and we look at a weighted graph that's coming from the prior knowledge, which has the weights of real values, and then we can calculate this score function of M and G, which is basically, it measures how similar M and G are. It's summing up the product of the edge weights for the 2 graphs. So we can see if if the motif has the has activation on all the positive weight at G, then this score is going to be higher. So it shows the motif and the graph are more consistent when the score is higher. And if we have a set of graphs, then we can calculate this weighted score function, which because of the linearity of the score function here, it's equivalent to do this for an average graph from the prior knowledge given a weight, given the weights for the average. To take an example, we look at these following 6 graphs from different sources. The first 4, A, B, C, D, are from the literature. The first one is for generic plant, circadian clock, and the B, C, D are for Arabidopsis. And the last 2 are from our soybean experiments. The E is the predicted network from COSNET. And F is based on the protoplast system. And given these graphs, we can, so we assume there's a weight associated with the 6 graphs. We're assigning a smaller weight for the older models or the generic models, and a slightly higher weight for the Arabidopsis models, and even higher weight for our soybean experiments. And then what we get as the average graph G bar is like this. Then we can calculate the score based on this average G bar for each motif. For example, here, if we're looking at this particular motif of LCL and PR7 2-gene negative feedback loop, then because in the average graph, we have the weighted edge weight of a 0.31 for the activation, and negative 0.38 for the repression. They happen to be the same sign as our motif. Then when you score the motif, you get a 0.69 as the average score based on the six graphs from different sources. Then by doing this for every pair of 2 or 3 feedback loop genes, we can pick out pick out the top scores, the top score networks. Then so this is the way one way to incorporate the information from both the literature and as experiments, as well as our COSNET prediction to come up with the motifs in the clock network. And for the ODE modeling, we have just argued that it's hard to do ODE modeling because of the complexity. But ODE models are still valuable because they are more biologically plausible, and they are generative model that can be used to predict the expression levels. So given a graph, we can always construct ODE models using some of the popular models like the G9-Weaver models or the more recent distributed delay models. And one important thing is to actually have enough data so we can do testing on the constructed ODE models. So we would like to have RNA-seq time series data and divide it into 3 parts. The first part can be used for graph learning using COSNET. The second part can be used to estimate the parameters in the ODE model, and third part can be used for testing the prediction power or the generalization of the ODE models. One example is based, so for this predicted repressor motif, we can write out the ODE model based on the distributed delay model in the paper by Takuda and others. So then if we have enough data, we can try to fit the parameters. So to summarize, there's this old saying that all models are wrong, some are useful, and but useful can be very subjective. It might be useful for one area, but not for the other. So what we argue is that we can more objectively say some models are simpler than others. So for our case, we can see that the graph models are simpler than the ODE models, and the motif networks are simpler than the whole entire Circadian Clock networks. And by focusing more on the simpler models, we hope that it can actually give us more insight, and it can actually we can actually get something useful from the very limited data that we have. And another saying is standing on the shoulders of giants. So we wanted to make use of all the information that we have, including all the prior knowledge from different sources, and that's what the motif mapping is trying to do. It's trying to take everything from different sources and summarize that by an average graph. And next steps, we would like to have more data, of course, and for graph inference, that could mean we want to verify the direct interactions experimentally, but we also would like to have more high quality time series data, which can be useful for both COSNET prediction and for the ODE parameter estimation. And given that we would like to modify our model to get better models. In particular, we want to be able to discover not just the 2 or 3 gene motifs, but the larger motifs, which actually gives you the pathway of the circadian clock. So these 2 steps are, of course, not isolated or in particular order. I would like to think that the biologists would do the experiments in a day and gather the data, and the modelers would look at the data and work on the models at night, and they can give the results of the model back to the biologist, which can be useful for experimental design for the next day. So by having enough of these circadian cycles, I hope that one day we are able to crack the circadian clock network. And that's my last slide with some references.
Excellent. Thank you very much for wonderful presentations. I am going to go ahead and bring in our expert panel as well now. So please do feel free, expert panel, as I am spotlighting you, to jump right in and start asking questions. Let me do that. I believe we have Carlos, Alexandra, and Elizabeth. Yeah, I see you. Here you go. And Alexandra, I see you. I'm coming. Sorry, I'm not as quick as Malachi. And Carlos. There you go. The floor is yours.
Well, thank you for a very interesting talk. I liked that you highlighted that there's more than just using ODEs to model a problem, and that before you start writing ODEs, you know, with the issues of parameter identification, etc., and what functional forms to use, you first have to identify the network. So, but my question for you is, the graph doesn't really tell you any sort of dynamic information, what happens. So before you use like CosNet or something, not CosNet, but the ODE, whatever package you use to write the ODE model that you then solve, have you considered something like a Boolean network where you say, you know, this and that? It gives you some dynamic information, but does not require you to know all the parameters and things like that, where you have and or statements that you use to predict what would happen.
I can answer that. The answer is no.
Okay.
We didn't consider coming up with Bernoulli models, but for the graphical models, we often look at sort of the linear Gaussian case rather than going to the nonlinear ODE models with all the McCallis-Mitten and Hill parameters and all that. So, so it's.
And are you getting periodic behavior in the ODE models? Because like just by looking at a network, I will not say, oh, that's an oscillator.
No, no, we've not really spent a lot of time looking at solutions of the ODE models because we don't really have confidence that we can identify them. But if we, in the graph, if we can see a loop where there's an odd number of repressions, so we know that it's possible to be oscillating. It depends on the weights that you assign with those. And then if you have a very complicated network, all it takes is a cycle in there with an odd number of repressions, and you can get an oscillation.
Thank you.
Can I ask a question about the weights that you assigned? So did I understand that those essentially act as kind of a how much do you trust that network? Is that so? It essentially seems like it's almost playing the role of some sort of Bayesian prior. Is that? Yeah. So, and I guess the other comment is, when you show kind of the results of the inference, and I assume there's kind of, there's the picture of what the result looks like, but I assume there's also, there's some level of sort of accuracy or weights or confidence in the various interactions that you infer. So have you thought about sort of putting this into some kind of Bayesian framework? Is that a possibility, or is that something that would make sense here? Or
so I guess I can answer. I think we're doing this exactly thinking of it as a Bayesian model. If you assign the same weight, the weight of maybe 1 for all the graphs, then the score function is basically like a likelihood function, assuming that the motif is the true network. So that's basically how the probability of seeing those different graphs given the hypothesis of the motif. And with the weight, that's basically doing not a likelihood, but it's based on the prior distribution given by the weight. So with the weight, that's exactly a Bayesian inference. At least that's the intuition of the motif mapping method. But we haven't looked at the performance in a more analytical way. So right now it's all heuristic.
Mm-hmm.
So just to follow up on that, so first of all, and this is my question is more, I guess, for Yoshie. Do you want to understand the kind of the dynamics of the process, as well as the kind of the structures that give you that these models are giving you, or what is it that would be like ideal for you in terms of understanding the system from modeling perspective?
Great question. The first step is to understand the structure, because we don't know it.
Mm-hmm, mm-hmm.
And we need to know the structure to be able to really see the dynamics of the network.
Correct.
And we certainly want to know the dynamics.
Right. So when I saw, so by the way, Xiaohan, you did really cool modeling. But one of the things that I was curious about was exactly what Elizabeth was saying, that if you have so you have the data, and you have what I'm getting is that you want to understand the structure of the system to then tie it to a certain set of dynamic behaviors. And so to me, that says, well, you have prior data and you have multiple hypotheses. And if you have a Bayesian formalism and you're really calling this a likelihood, then you could, in theory, should be able to capture or to calculate the evidence for all your different structural hypotheses. And so if you calculate the Bayesian evidence, you would be able to do kind of a multi-model inference to rank the models given your data. Is that kind of where you're going, or did I don't know if I'm understanding. And I guess I'm asking Xiaohan and Bruce.
Yeah, I like to think that's similar to what we are doing, but yeah, we haven't really seen a Bayesian framework used for our case, but I believe there must be many works out there. But I agree that that's the Bayesian inference would be a very natural way, and that's basically what we have in mind.
So the really cool way of doing it, so there's some, I don't know if you do Python, but for everyone that's listening, Python is the future. You should all learn Python. But if you're doing Python, there's a really cool tool called Multinest, PyMultinest, that allows you to actually explore the Bayesian evidence of your model. Julia's only for number crunching, Elizabeth. Yes, Julia's okay too. I will accept Julia. But anyway, Multinest allows you to actually calculate the model evidence and infer the model parameters, whether you're interested in them or not. The other thing that I was saying was that I would like to add is, in all these systems, you're going to have unidentifiability in your parameters. But one of the things that we see in our work is that even if you have parameter unidentifiability in your very large networks, when you look at when you actually calibrate your model to the data using Bayesian optimization, Bayesian inference, and you look at the dynamics of the network, it turns out that the structure of the network and the parameter values limit the number of execution modes that can travel through that network. And that's something that we actually have seen in many networks. And so I don't know if you're very familiar with dynamic analysis, but what that would enable you to do is to say, okay, given my network, these are the execution modes that are possible in this structure. How do they relate to the data that Yoshie is collecting to eliminate the models that don't make sense from a dynamics perspective? So I think that given the data that you're collecting, you have such an opportunity to really link these models and explore the hypothesis space to link that to the data and really accelerate that kind of knowledge acquisition. I'm sorry I'm taking over, Alexandra, but I just feel very strong about all these things, so I get really excited.
Can you spell the software that you mentioned again?
The software, and if you want to drop me an email, that's fine. I'm happy to help you guys and follow up. It's Multinest is one of the ones we used. PyDream, which we developed in my lab, is another one that could be really useful because this parameter inferences or network inferences processes are very computer intensive. So we've actually written a lot of these things to use GPUs. So if you want to drop me an email, I'd be happy to guide you guys or to at least tell you what not to do.
Yeah, thank you. That's very interesting.
I had a question about, I thought it was really interesting, the temperature data that you showed. And I think if I understood, you sort of have networks inferences that differ to a certain extent based on the different temperatures. And I guess I would very naively think that if one of these inferred interactions was reflecting sort of a true direct interaction, like the kind of thing you would go and then find with ChIP-seq, that might be relatively temperature insensitive. That may be the wrong assumption, but can you use any of that kind of temperature sensitivity to actually help you interpret the results of the network inference?
I'll say something. I think if you think about the differential equation models, if you have different concentrations, which you might have in different parts of the plant, you'll be in different parts of this phase space. Yeah. And you can linearize the model around the concentration levels, and in different phases in this nonlinear space, you're going to get different graphs. Mm-hmm. And it's the same genome as in all different parts of the plants, but they're differentiated by the expression level. So mm-hmm. I think it's a complicating factor mm-hmm. That I think really exists, which makes it harder to infer the network, because there isn't the network that's maybe the differential equations are closer to the truth, but then there's different networks, which we think of as linearizations of the nonlinear behavior of the plants.
Okay. Yeah, because I noticed you were using sort of robustness of the inferences with different algorithms. Seemed like it was a consideration, right? Like you sort of looked at these different algorithms had some disagreement, but also some agreement. And so it seemed like the interpretation was it's more like, we're more like, we trust more the ones that are sort of robust to the algorithm. So I was kind of wondering if you could do something similar with some of the biological perturbations, like, the robustness to various perturbations in the lab. But it sounds like maybe you're saying that that's very risky because of the
might be different numbers.
Yeah.
Well, it's also this is a more fundamental question to follow up on what Bruce just said. The kind of the stability of the networks and kind of this network space is something that is still kind of up for questions, right? Waddington is a pretty picture to look at it. But when you look at the rigorous aspects of it, there's going to be some things that don't quite match, right? Or you can't quite follow. Like, for example, if you apply the Waddington landscape to a steady state, to a limit cycle, you would have to be both minimizing and evolving in the cycle at the same time. So your differential for that process would be not defined, and that gives you kind of this weirdness especially when you have loops. So it's something to consider as well, as you guys move forward. I don't know if that made sense, but I can make it more clear if you want me to. Anyway. This project is really, there's a lot of potential. I'm very excited. It excites me to think about this thing.
All right, thank you.
Can the audience ask questions, or is that not okay?
You can, and I was just gonna chip in, oh, my camera just went out.
Right.
There are a few questions on your page as well. And you have time to answer about one more. And I do see some people have been leaving comments, so thank you. I'm not sure which one you'd like to grab.
I think we touched on most of them, but the last one is, do you have any concerns on potentially discarding all the motifs that you have not discarded otherwise if you would have considered more dynamics, Hill functions of species interactions? Xiaohan?
Any takers on that question?
I don't know. I guess maybe the question is, how many motifs should you save, or do you get the right motifs? Good question. The data is very noisy, so I wouldn't want to swear that these are the only motifs that are in the network. And but at least for a clock, we know that to have the clock tick, there has to be something there. So
maybe I'll. Oh.
Yeah. Go ahead.
Actually, wait. Were you going to ask a question, Eric?
But I don't want to cause trouble.
Go ahead. Go ahead.
So, okay. No, it's sort of related to what you all were saying, which is just that one of the things that fascinated me about that average graph is it's average over things that are all different. So they're not all the same, but they're all about circadian rhythm in plants. So I was just curious, like, why are they different? Like, so soybean's going to be a little different from Arabidopsis. Is there any insight that these structural things that so one way to look at this is just that the universe has some kind of structure in soybeans and in Arabidopsis and whatever. It's just the structure that it is. But if Arabidopsis has a perfectly functional circadian clock, why wouldn't we have the first hypothesis that it would be a conserved object across all plants? Flowering plants are actually pretty recent, right? I mean, not insanely so, but the idea that they would all have their own little clock with its own unique structure is kind of interesting, because it would suggest that they're trying to optimize or evolve for different things. So I don't know if any of your analysis of those different graphs or the graph you see in soybean, can your work kind of speak to that, or is it not able at a purely sort of structural level to start answering some of those questions?
I would say we don't know, and those are different papers that are trying to reconstruct the same clock. And I put it up to, we really don't know what the clock is. I think there's so much so much inference on so little data that it's not surprising to me that these different models end up being different. But in looking for commonness in them, we were hoping to come up with some simple pieces where there's a clock in there. And if biology could have 2 genes or 3 genes making a clock, that would be okay. But biology it's way complicated. There's probably 5 different clocks and different variations and ways they interact. So I don't expect it to be so simple to just rely on 2 or 3 genes for the clocks in all the different plants. And most of these papers have 8 or 10 or 15 genes in their clock network, and then you can't tell. I mean, why would it even tick? It can't even tell by looking at it.
But I don't know much about plants, but just comparing to the our circadian clock, do you see enrichment in the same motifs? Is it roughly the same motifs would appear, or is it totally different? I mean, obviously we'll need some negative feedback to give oscillations, but what do you see?
Xiaohan can answer that. I mean, most of those models do have some cycles with negative feedback.
Yeah, I think like one of more recent paper, they have 4 different negative loops. So I think any of the 4 would give you some oscillation. But they argue that maybe the 4 together would give you some kind of robustness, or so I think it should be of some function to have more than just a simple clock.
Interesting. Yeah, well, I think we have to wrap up, but the robustness is a great suggestion, and we would love to explore ideal robustness a little further. Thank you.
Excellent. Thank you so much, everyone. Thank you for a wonderful presentation and a great conversation afterwards. Thank you. A round of applause and wild emojis from the group to say their thanks. We are going to softly wrap for the day, but what that means is that we have lightning talks up next in 9 minutes. We have 3 really cool lightning talks lined up for you. So I just want to remind you, we have lightning talks today. We have another 2 lightning talks tomorrow. So hopefully your brain is not completely full yet, and you are going to stick around and hear a couple of people give short 10-minute presentations and then interact with them this afternoon and in the morning. I don't think Dave mentioned he did not have closing comments. He's going to go back to the organizer corner in the morning. So with that, I would actually recommend that maybe everyone stands up for a minute, takes a deep breath, woosahs, and then come on back here for those lightning talks starting in about 8 minutes. So thank you very much for a wonderful day today and a great kind of cap to the end with that last project presentation. So thank you. We'll see you back here soon.